Okay, thank you, Dudu. Good afternoon, everyone. My name is Esitungibi, and I'll be your chair for this National Biodiversity Assessment Webinar Series 8. Thank you for joining, and I hope you enjoy. For this webinar, I will give a short overview of what the NBA is, and then I will hand over to Davidine, Robin, and Betsy as the main speaker for this topic. Please allow me to switch off my camera because I have network problems on my side. The National Biodiversity Assessment is a very complex and covers many aspects of biodiversity. It has assessed 3 levels, which are ecosystem, species, and more recently, the genetic diversity. Today, we're only going to be presenting on the species, specifically microinvertebrates. The NBA efforts, they built on the wealth of publication and existing resources, it has resulted in a range of NBA products, primarily a book that you can download, and for the very first time on the NBA, and a website with 108 web pages and thousands of images, plots, and maps. Also, the regular updates of the NBA via these websites are now possible by the NBA team themselves. Also, the data science skills that we have built over the four years have enabled us to take a significant step towards the Living National Biodiversity Assessment. And we will use this momentum to improve our biodiversity monitoring systems more broadly. And of course, all of this required constant innovation and problem solving with few global examples to follow. It also requires collaboration with both in South Africa and with international scientists and institutes. This brings us to really key aspects of the NBA, which is significantly undertaken. The NBA was the collaborative effort from more than 490 contributors, representing approximately 110 institutes. It also took approximately 23,000 person days of work over the past 6 years. And it requires strong support from the leadership team and the willingness of our partners to help make it a useful product and process. So thank you all for being involved. While taxonomic capacity is lacking for all groups, the situation is particularly dire for invertebrates. Foundation data for invertebrates are urgently needed, specifically those with high levels of endemism in South Africa. Initial priorities include work on important terrestrial invertebrate pollinators, freshwater invertebrates that are sensitive to water quality change, endemic estuarine invertebrates, and endemic marine invertebrates. These are the gaps that were identified in the NBA 2018, and today we're presenting some of the taxon groups included in the National Biodiversity 25 that are addressing these gaps. So I'm now going to hand over to our main speakers today. Our first speaker is Davidine, who is a senior scientist, Threatened Species Program, and co-chair of the South African Species Specialist Group. Over to you, Davidine. Thank you.
Thanks, Asetu. Next slide. So today I'll just give a quick overview of the state of our freshwater crabs. As Asetu has mentioned, the NBA has gone online, and for all of our species groups, there's a species page covering information and summarizing the results for each of the taxonomic groups. Our freshwater crab page is coming soon, so keep checking back on that NBA website so that you can learn more about the overall results. In 2008, our freshwater crabs was first assessed as part of the Southern African Freshwater Species Assessment. And in the early 2000s, there was only about 14 species known in South Africa. And in recent years, through active taxonomic work, 24 species has been described. And this is from increase in field surveys within gap areas. Can you just click SA2? Of our freshwater crabs, the 24 species that have been assessed, so all of these species have been assessed globally and is available on the IUCN Red List. Only 3 species have been classified as threatened. These are 3 newly described species in the last 3-4 years, and these 3 occur in highly restricted areas within forest patches, and has not been found outside of those forest patches yet. So obviously new species, and we're hoping for further exploration. And with new information, it might change the species status in future. We click again, SA2. Main pressures for those 3 crabs have been indirect from firewood collections within forests, so their habitat being impacted by activities, and then also habitat loss and degradation due to agricultural activities within these forest patches. Next. As I've mentioned, lots of active taxonomic work within this group, lots of new species discovered, and really showing the value also of citizen science, where Prof. Saville Daniels, the taxon lead for this group, with support from Dr. Nasreen Peer, both based at the University of Stellenbosch, have also illustrated the usefulness of citizen scientists in discovering new species and also engaging with the platform iNaturalist to review observations and actually identifying species that are possibly new to science. Just click again, SA2. Some of these knowledge gaps for freshwater crabs have also been filled by projects like the FBIP Large Grant Refresh project, and some of the other groups that you'll hear about now has also benefited from that effort. For freshwater crabs, the way forward is understanding species ecology and how these species are interacting with pressures. There's noted some resilience for these species, but we have no understanding of their threshold. There's also no population level of information available for this group. So there's lots of research still to be done. And it's really just great to see the activity for freshwater crabs across South Africa. Thank you. I see you're muted.
Sorry, Dudun. Thank you, Dudun, for such an informative talk. Now I'm gonna hand over to our second speaker, which is Betsy. Betsy is a research associate at Rhodes University and Sion, and she's also a co-taxon lead for the large branchiopods. Over to you, Betsy. I think now I'm gonna have to stop sharing my screen, right, Dudun?
That's correct. Betsy, you can start sharing.
I'm not able to share at the moment. It's still blanked out. If somebody can just maybe give me permission.
Okay. Robin, can you see if you have the same issue? Betsy,
Yes,
could you
I
maybe...
do.
Oh, gosh. Okay. Betsy, can you maybe just email me your presentation just in case Samkita, I think this is your meeting, so you need to be able to give access. Sorry, I think today we're just having lots of gremlins.
Okay, there you go.
Lovely.
I have permission.
Awesome.
Let me just quickly share. Can you see my screen?
Yes.
Okay. So yes, my name is Betsy and I'm very excited to share the results for the first National Biodiversity Assessment for South Africa's Anastraka. If anyone doesn't know yet what Anastraka is, it's a group of freshwater brachiopods, and South Africa is a global diversity hotspot. with about 60 currently described species in the orders Notostraca, Anostraca, as well as Lavicaudata and Spinacaudata. And of these groups, Anostraca is the most diverse. Currently, there's about 43 species that we know of in South Africa. And then these groups of organisms actually occur in quite a diverse range of habitats, anything from salt pans, bare rocky pans, small isolated pools, grassy pans, typical small depressions that you would find in KwaZulu-Natal, ephemeral wetlands in the Highveld of Mpumalanga, and animal wallows as well as rock pools. So it's quite a diverse range of habitats. That's only a few to mention, but what they all have in common is the fact that they are all ephemeral habitats. So they're temporary, they only last a very, very short time. And these organisms are very well adapted to live in these ephemeral habitats. Their eggs go dormant during unfavorable environmental conditions, and their eggs are also very tolerant of heat and drought for prolonged periods. But more specifically, they're very dependent on this temporary state, because the temporary state is very important for environmental cue for them to actually complete their life cycle. So you can't go and make their habitats permanent. You need the habitats to actually stay temporary. More specifically, the Anostraca are very strong habitat specialists. There was a study done by Christopher Rogers in America that found that most of the Anostracan species occurred only in habitats with specific geochemical ranges that are attributed to specific species. So you can imagine, with this incredible amount of diverse habitat types, when these specialized species are so strongly specialized, there's also an opportunity for high biodiversity as well as high endemism to occur. Now, the reason that we used the focus of Anostraca on for the NBA is the fact that they are the most diverse group. They are strong habitat specialists, but they are also very well taxonomically defined, which makes them perfect indicator taxons for these temporary freshwater habitats. To give you an idea of what the red lists were, the red list assessments were like in the past, in the beginning of last year, only 30 species from about 353 global species were listed on the IUCN red list. And from that, 4 was from South Africa, which was mainly work done by Michelle Hamer in the 90s. Only 4 species at that time met the criteria to be officially published on the Red List Assessment website. But considerable advances have been made in Anostraca research in South Africa since. And that is why we were able to attempt a proper assessment of the taxon group for the 2025 NBA assessment. Now, we focused on mainly the native South African species, although some of them do occur in the sub-region as well. They are primarily considered native to South Africa. And a lot of the work, as Devadine mentioned before, was made possible by the FPIP Refresh project. If you are familiar with the NBA website, these stats might look a little bit different, because at the end of 2025, the published results of the NBA was for 36 species, but we since actually worked on another 6 species. So we have 42 species at the moment. And of all those 42 species, 60% are currently widespread and abundant, which translates to being least concerned, while 28 of our species have a high risk of extinction, and then also that's our threatened species. 2 of these species are critically endangered and possibly extinct. And then we have 40% of our species that are endemic to South Africa. If we look at the Red List Index, obviously this was the first time the Red List Index for Anostraca was calculated. So there's no specific trend. But if you look at the aggregate for all taxonomic groups in South Africa, the Anostraca falls well below that line. And another freshwater taxon group is the fish that also falls below that line, which translates to having high levels of threat, and many of them are actually at risk of extinction. And this correlates quite well with the fact that the other freshwater ecosystems are also highly threatened. Now, some of the pressures that our species are facing, I guess it's the same for all other taxon groups, is the fact that they are being threatened by habitat loss and habitat degradation that is caused by residential and commercial development, agriculture, as well as mining. And then if we look at protection level, it's just important to mention that this is not, we haven't done a protection level indicator analysis just yet. So this is just a representation of protected areas overlap. And 5 of our species are currently overlapping protected areas. Most of these are in the northern KwaZulu-Natal area, and then we've got one in the Meerkat National Park in the Northern Cape and one in the Eastern Cape in the Thomas Baines Nature Reserve. But this overlap alone is quite deceptive actually, because there's no specific conservation measures in place for Anastraka. And most of these protected areas are actually mandated to protect something else. So just to give you an example why this is so deceptive, the species that is found in the Thomas Baines Nature Reserve, it was originally collected there in 1989, the first time and the last time. So that was found there, and then just after they described it, that habitat was actually filled with water permanently to attract more birds to the nature reserve. Increased tourism, and as a result, unfortunately, the species has never been found since. So as I mentioned earlier, they do rely on temporary aquatic habitat, so they have specific conservation management actions that need to take place for them to survive in specific areas. So if you are going to pump their habitats full of water, they will disappear. And this is a very good example of how ineffective conservation areas is actually for the species. So that is why the protected level indicator analysis is very important to be done. So just a quick summary of our progress. We have increased quite a lot of the current published assessments on the IUCN Red List. Currently, South Africa makes up 58% of the global Anastraka IUCN assessments. We have already published 36, and then later this year we'll publish another 6. Unfortunately, our data is still severely fragmented. If you look at the number of records across the different 1-degree grid cells, there's still large gaps of areas that have never received any data, species data. And if we look at some of the habitat opportunities, only looking at depressional wetlands that are still in a fairly good condition, we're not considering rock pools along the gradient escarpment or any other of these elusive pools that are difficult to map. Just looking at depressional wetlands, you can see that there's still a very large opportunity to do more sampling and to gather more data, especially for the Northwest, Free State, Limpopo, and Mpumalanga province. We have also found that our data is quite biased. The diversity and the distribution patterns currently are very strongly influenced by sampling efforts. So this risks assigning inaccurate extinction categories. So very important to actually decide on a strategic way to sample these areas now going forward for the next NBA. Many of these gaps are actually caused by several challenges that we face when you work with Anastraka. Apart from inaccessible terrain, the unpredictable and short-lived aquatic habitat phases and the life history traits of the Anastraka make it quite difficult to sample them. Just to give you an example, here's 2 photos. One was taken in February 2020, and the other one was taken about 2-3 weeks ago in May. Both of these sampling efforts took place 3 weeks after the first inundation of that season. But you can see the habitat conditions were quite different. The first photo shows the water depth about just above my knees, and the water temperature at that time was 28 degrees. And we found at least 5 different species of brachiopods during that sampling. And then 3 weeks ago, the temperatures were 15 degrees and the water was about 60 centimeters deep. As you can see, it was quite deep. I only found tadpole shrimps and no Anastraka. So they're quite temperamental, and you don't always know when you're going to be able to find species to add to your data set. And then also there's inconsistent protocols for collection across variable habitat types. And all of this basically makes for very unreliable population estimates. And we definitely need more reliable population estimates to do IUCN red listings. We also have a scarcity of local taxonomic experts. So there is this risk of misidentification. Much of the research that has been done in the past also only has genus level records. So that doesn't really help us much in terms of biodiversity work. Then there's also a lack of dedicated funding for staff time. Most of the funding currently available that we receive is mainly for project costs, but doesn't necessarily include staff time. And there's no dedicated employed Anastraka specialist in South Africa at the moment. Priority actions going forward, we were very grateful to receive a small project grant from the Mohammed bin Zayed Species Conservation Fund to look for those critically endangered, possibly extinct species. So that's definitely on the cards for the next year. And then by March, hopefully March next year, we would be compiling an assessment protocol and guidelines for evaluating Anastrakan species for the DFFE screening tool. And before we can attempt any other red listing or by the NBA next biodiversity assessment, we definitely need to develop a grid-based sampling strategy to minimize that bias and improve our accuracy of our red list assessments. Now, how you can get involved. Again, iNaturalist is an incredible tool, not necessarily to take photos of the identifying features. That's quite difficult. As you can see on the right-hand side, we use very high magnification. These are 2 Streptocephalus species, and we use the projections and the different spines and things on their hand region to identify them. But if you are able to, if you do see them, you come across them, at least take photos of their face, or at least a nice clear photos of what they look like. A little bit of a description of their habitat, make sure that their GPS locations are accurate. Or if at all you are able to sample with permits, obviously, and you have these specimens, you are more than welcome to contact me. Thank you very much.
Setu, your mic's muted.
Thank you so much, Betsy. We enjoyed your talk. It was very informative. So now I'm going to be handing over to our third speaker, which is Robin Lyle. Robin Lyle is a researcher she's a researcher at the Agricultural Research Council, and she's also a taxon lead on spiders. Over to you, Robin.
Robin, your mic's
Sorry,
muted.
guys, I was muted. Murphy's Law. Sorry about that. Welcome, everyone. Thank you for listening to our talk today. I'm very excited to share some of the results that we have to show. My talk is not as long as our previous speaker, but we have got some interesting stuff to share. So this was a very long-term project. I can remember when I started at the ARC, which was before 2015. They already started with the red listing process, and the team has changed considerably. It is a collaborative project, obviously, with SAMBI, the Agricultural Research Council, the University of Venda, the University of Free State, and the National Museum of Bloemfontein, as well as a number of interns who have worked on it throughout the years. And we have Sma and Eugene, who worked at some point during this period on the red list assessments. And then the core components, you can see the core members on the other side. But I do have to just make a special mention to 2 very particular team members. The first is Dr. and Professor Ansi Dippenaar-Schoeman. This was her vision. This is the cumulative contribution of the work that she's done throughout her career. She's been retired now for about 10, 12 years, but she still publishes papers. She's still doing, well, she's cut down on identifications, but she still works on spiders almost every day. And if it wasn't for the work that she done and the work that she started, we would never have got to this process where we are now. Ansi's published numerous, over 100 papers, both nationally and internationally. She's supervised over 22 students. She's written various popular articles and popular books. She's really the mother of African arachnology. Spiders in South Africa would not be where they are now if it wasn't for her. Then the second special mention I need to give to is Professor Stefan Voort. He played a pivotal role in getting large parts of the red list assessments done, such as the area of occurrence and that type of stuff. He was also a very close friend to many of us in the spider community, as well as the students that he supervised. He was a lecturer at the University of Venda. He touched all of us in various different ways. I have some of my fondest memories is with Stefan at some of the African Arachnology Society colloquiums, after a couple of beers and some late night chats, and would range from personal to professional, and he was always there for all of us with a good laugh and a good joke. And he was just someone gone too soon. But both of these made the red list assessments that were included in the MBA possible, in very different and very unique ways. So I feel they need a special mention for the work they've done. So the next slide. So this is just the status of spiders in South Africa. All our red list assessments are based up until 2020. And at that time, that was about 2,200 species that were evaluated. And you can see there's a massive amount that are of least concern. We're quite lucky compared to the other taxa that were included. We do have a low number that are endangered, critically endangered, or vulnerable, but we have got a massive amount that are taxonomically or data deficient for taxonomic reasons. And you can see that in the light gray area. But what is very special about spiders is that we also have a large variety or large diversity of species that are rare, which is a South African level of assessment. You won't find on the international red list assessments. The major pressures on spiders is basically the change in landscape use, which is residential or commercial development, aquaculture and agriculture. It's quite funny because we do work at the Agricultural Research Council, but we have seen the importance that spiders also have got in agriculture. And they are also, there are smaller risks, but those are the two main pressures that are on spiders. Spiders overall are quite resilient, but there are some that have very specialized life cycles that are, where a small change in the habitat can greatly affect them and their population numbers. So as I mentioned before, the data deficient species are a huge number of them. And this number has changed since the red list assessments have been done because more species are described on a regular basis. But we had at the time 657 species that are data deficient. That means they're either known from one sex, either the male or the female, or there's not enough distribution data, or that nothing is really known about their life cycle. So currently, I think we're heading towards almost 3,000 species for Africa, but we still, or for South Africa, not Africa. Africa alone would be way more, but we are just at the tipberg of the species in Africa, and there's large amounts of gaps that need to be filled, whether it be finding the opposite sex of the species, understanding its life cycles, or even just understanding its full distribution range. So for this, we need to do ongoing taxonomy with ongoing sampling, and we need to start training and taxonomists, but we also need to get them employed, permanently employed, or at least on contracts. And as mentioned by the previous speaker, taxonomy is a scarce skill, but it's the primary or foundational science that needs to be in place for any other science to take place. Without a name, you have nothing. And again, we need to understand their life cycles. So there's so much more than just giving spider a name. We need to understand its biology and its ecology as well. So just to touch on the rare and endemic species, as I said, the more taxonomic work we do, we find out that we have more rare and endemic species. So just to refer to the Stasiomopus genus that was recently revised in the last few years by a student who's now doing a postdoc in France, she worked just on Stasiomopus species found in the Karoo biogap area, which is the area that was proposed for shale gas fracking. She found a number of new species as well as short-range local endemic species. So they're very localized, they have small distributions, and often they're very hard to find. This is not just to the megalomorphs, it applies to the uraniomorph species as well. And trapdoor and baboon spiders are often the focus for this because of how they move and how they find. The males have to leave their burrows to find a female, and the female literally waits for them to come. It's actually quite cool. I've done a field trip where I've seen the male literally knock on the trapdoor spider's female's door. So they have their own courtship rituals as well. So it's quite cool to see in real life. So as I get, spider diversity is increasing in South Africa due to the number of active taxonomists and students, but we still have a long, long way to go. And many of these students get their degrees, but they don't go on to find permanent jobs afterwards, which is a concern. And then how can you get involved? So through SAMI, there's our naturalist website. You're welcome to join up. There is a special Spider South African iNaturalist group. Dr. Antti Dippenoskum is very active, and she is so along with various members of the community. Many of her photos that she's putting up recently is also to a member of the public, Peter Webb, who made a massive contribution that is related actually back to specimens. So she's busy loading those up. He also passed away from cancer not too long ago. So please, if you want to join or contribute, iNaturalist Away, you're welcome to reach out to me at any point. And yes, thank you so much for your time. And I hope you enjoyed it.
Thank you so much, Robin. From now on, I will briefly touch on the current knowledge gaps. Oh, sorry, I have to share my screen first. Sorry, guys. Can you see my screen?
Yes.
Okay, thank you. Thank you, Robin, for the talk, and thank you all to our speakers. But then before that, from now, I will briefly touch on the current knowledge gaps. Today, we have seen how some of the knowledge gaps have been addressed for the invertebrates. However, there is still a lot of work that needs to be done to improve our taxonomic understanding of invertebrates in general, and in particular for our marine realm. In order to do this, we need continuous fundings for museums and other collection facilities is needed to ensure that we are able to monitor and report on the status of species. While the work for the citizen science are currently filling some of the gaps, these efforts need to be sustained. And also you can explore other knowledge gaps and priority actions on the NBA website, and we hope that these gaps will continue to be addressed. Thank you once again for the keynote speakers, Betsy, Robin, and Dudin. Let's please give them a round of applause. I think now we have like 3 minutes of also for key questions or input from you guys. And while you are thinking about that and putting up your hand, I just want to mention that each NBA builds on and contributes to a body of knowledge that enables a multitude of stakeholders to develop the various policy response and detailed actions required for the benefit in both diversity and people. As the Deputy Minister said at the launch event, the National Biodiversity Assessments give us the knowledge to act. Now it is up to us to turn this knowledge into lasting changing for nature and our people. Please, everyone, if you have any questions for our speaker. Oh, okay. I'm not sure if I can see who's raising the hand on my side, or I can just stop sharing from here so that I can see. Yes, Pumza.
Okay. Good afternoon, colleagues. My question is going to Dr. Pete, about the Anastroca species, because Dr. Pete is saying that this species was here in South Africa in 1989, and then it just disappeared. I wanted to know because it falls under the families of the shrimps. Is Anastroca the species of South Africa, or is an alien species here in South Africa? Because it just disappeared for a long period of time, and then it just appear again in around 2025. I wanted to know
I-
whether
Thank you for your question.
it's because
So
of
no,
climate change that
yes.
it come back to South Africa.
So
Yes.
it's not an alien species. There's only one alien species that we know of currently, which is called Artemia franciscana, and they mainly associated with the aquaculture, along the estuaries and so on, the salt works and things like that. The species that I presented on are all indigenous to South Africa. They were only found once ever because of the habitat that was destroyed. So we are trying now to go and look for them to see if we can find them in different habitats. I don't know if that answers your question.
Yes. Okay. Thank you. Thank you, Doc.
Yes, Kate.
Thank you. Also a question for Betsy. I was wondering the habitat for the Anastracans, can they survive where you have a permanently inundated body of water with temporarily inundated margins? So just from a management
Okay.
perspective, yeah, thanks.
So yeah, so the rule of thumb is that they want that desiccation, right? The only place where I actually found them is elephant wallows or areas where that's permanently drinking water. They can sometimes bring them in, if the elephants bring them in, and then the eggs were dry and from the mud or on their feet. And when they bring them into permanent water, get inundated, then they can hatch. Because I have found them in elephant wallows that are permanently wet. But my only explanation for that is maybe that the eggs were brought in through these vectors. They could maybe, if on the edge, if the area expands, maybe if there's an egg bank along the edge and it dries out for most of the year and only gets inundated for a specific time of the year. I don't have any physical evidence of this, but my thinking is that if it's at least temporary for a little bit so that it can dry out, if it gets inundated again, then I'm sure it will hatch if there are egg banks in that region of the habitat. That makes sense.
Thanks, Betsy.
Is there anyone with a question or have some inputs on the talks? Okay, I have one question for you, Betsy. Given that the NBA is currently assessing genetic diversity and that there is a significant taxonomic knowledge gaps for many species, are there any current projects, initiatives for future plans focusing on DNA approach such as barcoding to address these taxonomic gaps?
That's a very awesome question. Thank you, Setu. So we did, through this refresh project, we actually sent a lot of our data to the zoo. And I think Matlatse, I saw your name. I think you are in the audience. He's been doing our barcoding. We haven't done any publications or actually working on through the data to look at cryptic species, but we do think that there are certain species, especially those species that are quite widespread and that have quite a variety of them. Their morphology is a little bit different than the other. It looks like the same species, but we think they might be cryptic species that will only be shown through genetic analysis. And then recently, we've just described a new species, basically, thanks to genetic analysis. It's a Phyllocryptus species. It was called Phyllocryptus spinosa, and we have found now that it's different from the cosmopolitan Phyllocryptus species. So we little bit of genetic data, but we definitely still need to do quite a lot of that in future.
Thank you so much, Betsy. Alice?
Thank you. Also a question for Betsy. I'm very fascinated by this little creature that I hadn't heard of before today. Is it possible that some of the distributions will be affected by animals, like birds flying to pans, migrations and things like that? Is that something that's not yet known? I guess not, because we haven't got, a very full, clear picture of the distribution. Do you think that will have a big impact?
Yes, so I don't know of the impact of it. We do know that they actually are the main vectors. So studies have shown that wind can be a vector, but the water birds are specific, very good, strong vectors. There's also a study that showed that terrapins are a good vector of distributing some of these eggs, and then actually also animals, large mammals, as I said earlier, elephants. But water birds have been shown to be quite important vectors for distributing these eggs.
Thank you.
Any questions, anyone? Okay, I think we don't have any for now. Thank you once again to the main speakers and to the attendees. And also note that the recording of these webinars will be uploaded to the Sunbee YouTube channel, where you can share and like. Oh, thank you, everyone. I think I'm going to close now from here. I'm not sure if there is a question on the comment, or someone is. Oh no, it's not a question. Thank you,
It's
everyone.
not a question. It was just a comment of support to Bill. This is Mahlatz, to Betsy. This is Mahlatz working with Betsy on this Anastroca. We're trying to barcode them for her in combination with her identifying them, so that it can become easier to identify them through DNA barcoding. We're just putting that comment. Thanks.
Oh, all right. Thank you, Mahlatz, for that. Thank you, everyone.
Thank you.
Thank you. Bye.
Bye.
Thank you. Bye.
Bye-bye.